Welcome everybody to this webinar series. We're going to be speaking today about advanced topics in using the exabyte-io platform. And we've been working for a little while on the new implementation of a machine learning pipeline. And today, James is going to be demonstrating some of the ways that we can apply this new machine learning pipeline. And we'll consider two specific data sets, one for materials and one for chemistry, and explain how we can utilize tools like Jupyter Notebooks and the native pipeline that we have inside the platform to train machine learning models and then do predictions with these data sets. So without further ado, James, please take it from here.
Alrighty, thank you, Timur, for the awesome introduction there. So before we get into things, I just want to remind everyone that this webinar is going to be heavily focused on the Exabyte platform and what it can do. There are some general topics, but everyone here should be a registered Exabyte user to be able to follow along later on. Everyone here is going to receive a PDF of this presentation afterwards. And also this webinar is going to be recorded and made available online. More info at our documentation site here. And then one last thing before we jump into things, I like interactive webinars, so feel free to interrupt at any point with any questions that come up, and I'd be happy to answer them. There are some Zoom tools available, chat, raise hand, and Q&A down here. All right, and then at a high level, what we're going to be doing today, it's going to be a little bit bittersweet. I don't think that Perovskites are sweet though, so I guess that doesn't work. We're going to start out by trying to predict the bitterness of a chemical based on just knowing its smile string, so whether it's bitter or not. We'll be doing a little bit of discussion at first over what features we're choosing. Then we'll talk about how to run Jupyter Lab on the Exabyte platform to do some machine learning or prototyping. And then finally, we'll use XGBoost and Optuna to train a classifier. Next up, we'll predict the volume of a Perovskite using a fully automated machine learning solution. As opposed to here, where we are going to be doing a very sort of focused approach where we select features and then we know which model we want to use. Here, we don't know what features we want to use, so we just select a bunch of them, and then we fully automate the production of a machine learning pipeline using TPOT. And so this will be sort of the more hands-off approach to machine learning. And then finally, we'll close out with some Q&A. All right, so let's get into the bitterness portion, predicting bitterness of a molecule. So it turns out that when we eat or drink something like this delicious coffee, the pathway that ultimately results in our brain having the sensation that this thing tastes bitter, it starts at the TAS2R receptor family of proteins. So a molecule will bind to one of these receptors, which starts that whole process that ends in the sensation of bitterness. It's actually really easy to remember the bitter and sweet proteins, it turns out, or the receptors for those, is that they start with TAS. TAS1 is sweet, TAS2 is typically bitter. And so these bitterness receptors were actually discovered around 2003 in this paper here. And I really wanted to show an image of them. However, it turns out that we don't actually know the experimental structure of these just yet. Here's a recent review paper that kind of discusses what we know and what we don't know. It's really interesting. I'd recommend checking it out. And so in lieu of doing that, we'll look at a different type of protein. Here is an enzyme, and this is a very important enzyme. This is a hexokinase. But many of the concepts that are involved in this enzyme catalyzing a reaction, which is what enzymes typically do, many of these concepts will still apply to the receptor. So specifically this concept of a binding site. So here, this is sort of a cartoon drawing of what is going on over here. So in this image, we have an ATP molecule, so the block here, or this molecule, and we have a xylose molecule, which is this little six-membered ring right here, or this hexagon here. And proteins will typically have, if they interact with molecules, they'll typically have a binding site, and the molecule will come into the binding site, and the protein will actually close up a little bit around it as it kind of binds to the molecule. This is called the induced fit model of binding. And so In a more realistic drawing over here, we actually have the molecules up here. They come into these binding sites, and we see that the protein actually kind of moves a little bit inwards on this binding site. And so what's going on in this picture? What is this hexokinase doing? Well, it's kind of in the name. I keep clicking that. So it's in the name of the enzyme. So a kinase, typically a kinase will add a phosphate group to something, which makes sense because we have ATP here. It's adenosine triphosphate. It has 3 phosphate groups in it. So that's our source for phosphate and adding phosphate to something. And then hexo. Oh, we have a sugar out here. Sugars have our 6-membered, well, or oftentimes 6-membered rings. So that's what this enzyme is doing is it's adding a phosphate group to the xylose. And this would actually be one of the first steps in metabolizing and breaking down this sugar for energy. So hexokinases are actually very, very important because this is, they're very involved in the process of metabolizing sugars. And so it's probably more important than tasting whether something is bitter or not, because this is actually how we extract energy from sugars, or one way at least. So molecules, it turns out, can bind to proteins, right? This is the basis for tons of biochemistry research, is characterizing proteins and how things bind to them. And in order to bind to things, recall that there are these binding sites, right? These binding sites have different shapes, as we kind of allude to in this drawing over here. Or I guess they do, because this is from Wikipedia, this drawing right here. And so the molecules, the shape of the molecule is going to matter to how strongly it binds. So the size of the molecule matters because we can see that there is physically space that the molecule is taking up. So the size of it matters. The shape of the molecule matters because, again, it's fitting into a physical space. The charge distribution of the molecule matters. So how is it, like, is it polar? Is it non-polar? Because proteins will have polar and non-polar region, or I guess hydrophilic and lipophilic regions. H-bond receptors and donors in the molecule will matter because oftentimes those will play a role in binding, and many, many, many other things. Entire research papers can be built on characterizing these binding sites. So it's a very, very deep subject, and we're really only scratching the surface. The data set that we're going to use for this problem of predicting whether a molecule is bitter or sweet, this actually comes from Kaggle. It's got about a little under 5,000 entries. But it looks like this paper actually came from this paper here. It doesn't say it outright, but it's using the same data. And here's this paper here where they actually tried to assess different chemical fingerprints and different models for classifying whether a molecule is bitter or not, or whether it's sweet or not. It's a very interesting paper. I highly recommend checking it out. So in the data set that we get, we have a few columns. Excuse me. So we have the name of the molecule. We have the taste of the molecule. So this can be non-bitter, bitter, sweet, or tasteless. We have a reference to the data set it came from, because this contains data from multiple data sets. We have the smile string. We have a canonical smile string, which I'll discuss later. We have whether it's in the bitter domain, and then we have bitterness. So we're kind of in a bit of a problem here because there's like 3 different assessments for whether a molecule is bitter or not, and they seem to be kind of contradictory. So really what we're just going to do here is we're just going to take this as our source of truth in this data set, this taste thing here. And so that's just what we're going to use. And we're going to kind of ignore these columns out here. I'm just going to choose this as the column we're trying to predict. All right, so now we have our data set. We want to do some data science on this, but we need computational time, or we need computational power to be able to do that. And to do that, we'll actually be running this notebook on Exabyte. So as Tamir mentioned early on, we've actually put a lot of focus into improving our platform for data science. And one of the things that users can do is actually run Jupyter Notebook or Jupyter Lab on any of the queues on the platform. This is great for rapid prototyping because you can rerun cells, run through entire scripts in Jupyter. It's great. And so we actually have a pre-made notebook workflow on the platform. So if we go to the workflow bank and we search for Jupyter Notebook, we'll find this notebook owned by the curator's account. And this is the notebook workflow. So go ahead and copy this into the account. And this can actually be used to run a Jupyter Notebook on any node or on any cluster, including the GPU clusters on our platform. For the purposes of this webinar, we're just going to run on the AWS cluster with 1 node, 16 cores, and we're just going to give it a max wall time of an hour because we have to tell it how long after we start the notebook to close it down. So we're just going to give it 1 hour. All right, and so here's actually how we can spin up a Jupyter notebook. So starting here, I am logged in. The first thing I can do is that I can go to my Dropbox. The Jupyter notebook will have access to any file in my Dropbox. So here's my Dropbox, and I can go ahead and actually click the upload button. All right, go to the upload button here. I can find my bitterness notebook that will be hosted on our API examples repository after this webinar. I've uploaded that. Here's the BitterSweet dataset that I have uploaded. So I've got my notebook and I have my dataset. So next I can go ahead and create a brand new job. I can select that Jupyter Notebook workflow that's in my account by just searching for it. So I'll search for it, and then I can click on that. And then I can give the notebook a friendly name, like BitterSweet Webinar Test. So I've given it a name, and then I can go to the Compute tab. Do I? I do go to the Compute tab, okay. And then I can choose 16 cores. I can go ahead and choose 16 cores there. Give it an hour, and then I can just click the check mark to go ahead and put this job into the queue in a pre-submitted status. Then I can go ahead and run the job, and I can wait a couple of minutes for the notebook to spin up because we have to build up this compute node and get it prepped and ready to go. After a couple of minutes, the job's going to start, and we can tell the job has started and it's running by this little orange glow in the notebook here. It's colored orange. So if we start this, I have clicked the job and I've gone to the Workflows tab, and we see that the notebook unit is actually orange. So I can click on the notebook unit. Here we get the normal output that we would see if we were running Jupyter Notebook locally. Sometimes programs will print to this output here, so it's good to have it around. And then we can go ahead and click on Lab. We also offer Jupyter Notebooks. I just prefer Lab because it's a little bit newer. It just feels a little bit easier to use to me. And so we're going to use Jupyter Lab, and it's going to load up. After a couple of minutes, it'll load. Alrighty, cool. And so then we can open up our classification notebook. And so here, this is going to eventually be on our API examples repository. That's why there's so much description out of here. We're going to kind of skip that, but what we will do, however, is we'll talk about the things that we're going to be importing here and the things we're pip installing. So any shell command can be run by prepending it with an exclamation point here. So we're actually shelling out the pip command. And here we're just going to install basically any non-standard library that we need here. And we can just do that right at the start of the notebook here. So we're going to install NumPy, Matplotlib, Pandas, Scikit-Learn, so pretty standard. XGBoost is a gradient boosting library. Optuna is a hyperparameter optimization library. We'll talk about what that means in a couple of slides. Imbalanced-Learn is useful for dealing with imbalanced data sets. It has some upsampling techniques. And then RDKit is a very useful chemical library that will generate descriptors for molecules. So I went ahead and I've made sure that all of those packages are installed before I'm doing anything. It'll take a couple of minutes for pip to install. And then we can go ahead and we can import everything that we need for the notebook at the start. It's a good idea to put imports kind of at the beginning of a script or a program, just so we know what's in here. That'll take a second to install and import. And then once that imports, here we go, we can go ahead and scroll down and we can read in this data set. We're going to be using Pandas for this. Pandas is a very convenient way of manipulating data that can be represented as a table in Python. It's got these convenient read CSV functions where we're just going to go ahead and read in the bitterness here. Here we go. So it's in the Dropbox, so we don't need to give it a path. We just give it the name of the file. We can go ahead and print out some value counts out here in the taste column, because this is the truth we're going to be using is this column. We can see that there are many sweet molecules, plenty of other bitter ones, a fewer tasteless molecules, and some non-bitter molecules. And we can print out the data set here that we showed on a couple of slides ago. Okay, and so we've read in our data set and we're ready to do some machine learning on it. The next step in the process is typically to figure out what features might be relevant to the protein binding. And here we're going to use a little bit of chemical intuition. So recall that the bitter taste is caused ultimately by the ligand-protein interaction, so a molecule interacting with a receptor. So what might be important to that? Well, things that cause intermolecular forces or influence them. So for example, proteins can have lipophilic and hydrophilic regions. And so aromatic rings, for example, they're very lipophilic, they're typically very greasy. And so it's probably a good idea to count the number of aromatic rings, just as an idea of maybe something that might matter. Here's an aromatic ring in this benzoic acid, a little benzene ring. We have the number of H-bond donors and acceptors in the molecule. Just as a recap, an H-bond donor will donate a hydrogen to the bond, and an acceptor is a lone pair on an electronegative atom like oxygen. So we have 1, 2, and 3 H-bond donors in this molecule, or H-bond acceptors in this molecule, and 1 H-bond donor. So that might matter because that's going to be sort of related to the hydrophilicity of the molecule, and H-bonds oftentimes can matter to these protein binding sites. We also have the molecular weight of the molecule. This will help give a descriptor of the size of the molecule. We have the largest and smallest partial charge in here that was estimated by RDKit, which again might matter because polarizability will matter to, or how polarized the molecule will help matter to its binding. And then we have Morgan fingerprint densities. I'm not going to go too deep into what a Morgan fingerprint is in this webinar. Here's a couple of articles that go into a lot of detail about them. But essentially what they are is they are descriptors where there's a vector array of bits, and different features in the molecule will turn on and off certain bits. And in RDKit, it actually has this fingerprint density for Morgan fingerprints, which will take the sum of all of those ones and zeros, and it divides it by the number of atoms in the molecule. So conceptually, it can be thought of kind of as how many features, how many chemical features are in a molecule, and then sort of like the density of that kind of. So that's sort of what this is doing. So it's a little bit less interpretable than the other ones, but it may be important to include. All right, so now that we have our features, we can actually go ahead and do our feature engineering with RDKit. So the first thing that we want to do before we do that is we want to process the data set. And that's what we're doing out here in this cell. So what we do is we go ahead and pull the name and the canonical smile string. We're pulling the canonical smile string because the goal of canonical smile strings is to make it so that one smile string represents one molecule. And so this will help us detect duplicate molecules. And then the name, just because it helps us actually understand what molecule we have, because it's a little bit more readable than a smile string. So this is like fructose, for example, which I wouldn't be able to tell by just looking at the smile string. And then we're going to say, here's some classes in the molecule, or here's some classes in the data set. So we have sweet, bitter, tasteless, or non-bitter. And this bit of code out here is essentially going to do this all versus one approach, where we're going to make this bitter column here. It's going to be 0 if it falls out, if it's not bitter, not non-bitter, because that's one of the columns. But if it's not classified as bitter, it's going to be 0. And if it is classified as bitter, we're going to give it a 1. And so that's what we're going to do to turn this for a class classification problem into a binary classification problem, which is oftentimes easier. And then we're just going to put that into the bitter column, and then we'll go ahead and try to drop any missing rows and any duplicate entries in our data set. And so that's what that's going to do. And then we're going to go down to RDKit, or we're going to run some RDKit code, and we're going to use this Lipinski set of descriptors from RDKit, the descriptors submodule as well, to generate those descriptors we were talking about earlier. Excuse me. And then when we're doing this, by the way, in this data set, many of the smile strings are actually invalid in the sense that RDKit is unable to process them. There might be like a closing bracket missing or something. So we're going to actually surround that by this try except block out here, where we're going to say, go ahead and try to make the descriptors out of the smile string, but if something breaks, just put a none entry in here. This way we can just go through the entire data set, ignore any errors, but mark where we had errors. And then we'll go ahead and drop any rows that we were unable to actually featureize in the data set. And so we go ahead and do that. We can see plenty of errors as RDKit walks through the data set and generates those features down here. All right, and then one last thing before we jump into the machine learning part is we're going to do a training and testing split. So a train test split's very important in helping to determine whether a model will generalize or not. So we'll go ahead and do that here using Scikit-Learn. We'll go ahead and create our X and Y, our sets of X and Y we're predicting on, and then we'll use SMOTE. I'm not going to go too deep into what SMOTE is, but it's essentially an upsampling technique. This is a variation of SMOTE, and what it's doing is it's helping to solve this imbalanced classification problem. So for example, here, before we resample, we have 1,300 non-bitter and 800 bitter molecules. So by randomly guessing, if we just randomly guessed that it was never bitter, we would be right about 2/3 of the time, which is a bit of a problem. And so upsampling will actually create new synthetic examples, so sort of new fake entries for bitter, so that the model has something to train on, and we don't have this imbalanced classification problem, so that random guessing is closer to 50% wrong. So that's what that's doing. Okay, so now we have a training set and a testing set. We've upsampled our training set, and we're ready to do some machine learning. What are we going to do? We're going to actually use XGBoost in this example. So it stands for Extreme Gradient Boosting. XGBoost is a popular open source library for gradient boosting. It's available in C++, Java, Python, and many other languages. It's extremely fast and efficient. That's why it's so popular. It was released in 2014. The paper was a couple of years after that. And just to sort of talk about how popular this is, going to Google Scholar, there are over 10,000 citations for XGBoost. The 2015 conference paper has like 4,000 citations alone. So clearly a quite heavily cited package. Here's a couple of links down here. It's an implementation of gradient boosting without going into too much detail for the interest of time of this algorithm. It trains on multiple rounds where every round we add a new predictor, in this case, a new decision tree to a whole group of decision trees. And then we sort of add trees trying to minimize the gradient of the error with respect to adding a new predictor. It's very interesting, and it's actually explained on the XGBoost site. But because this is sort of a complicated model, there's many hyperparameters to tune. But so because there's a lot of hyperparameters to tune, we need a strategy to tune them. So a hyperparameter is sort of like a knob that we can tune on the molecule or on the on the model. So this is different than a parameter. So a parameter is something that we fit to a data set. So for example, in linear regression, the coefficients in a linear regression are parameters. A hyperparameter is something more fundamental to the model and how it will be trained. For example, if we now add a penalty term to our linear regression that's going to penalize the squared sum of the weights, so to prevent it from like relying too much on one feature over another, we would get something called ridge regression. And so then we would sort of have to decide how much do we want to penalize the squared sum of the weights of the model. The penalty, how much we penalize it, would be a hyperparameter. So we don't directly fit it to the data, but it affects how we're training the model. Another example might be in a group of trees predicting something, how many trees are in the ensemble. And so that's sort of what a hyperparameter is. It's sort of like at a higher level than just fitting to the data. And typically, what we want to do is we want to tune the hyperparameter to try to minimize the error of the model. So we sort of indirectly train them on the training data. Unfortunately, though, there's actually potentially a lot of hyperparameters that we might want to look at. So for example, if we have like 5 hyperparameters and 5 other hyperparameters, the simplest solution would just be a brute force search called grid search, where we physically would walk over the grid of every single possible combination of hyperparameters. That's why it's called grid search. This is brute force, and it's going to be like n times n times n times however many different hyperparameters that are that we're looking at. So it can blow up in computational complexity pretty quickly, and it can take a long time. As a result, oftentimes a random search will be employed instead. So we don't sample the whole space, we'll sample a subset of it and hope that we get lucky and get a good set of hyperparameters. And a sort of more refined way of doing random search is a so-called Bayesian search, where the hyperparameter optimizer, in this case Optuna, it's going to randomly sample groups of hyperparameters. And if one set of hyperparameters is performing better than the others, it's going to sample similar sets of hyperparameters. So sort of a more intelligent random search is what Optuna is going to be doing. It's very easy to use Optuna, despite the sort of complexity and what it's doing. It's easy to use it. Here's a 10-line example from the Optuna website where we can import Optuna. We can define our objective function and give it a trial. This trial is supplied by Optuna. And essentially in this trial, the purpose of it is to pull from a random distribution that's defining our hyperparameters. So here, X might be a hyperparameter, and it's going to pull from the uniform distribution from -10 to 10. And it's going to return this value here, which is just x minus 2 squared. And I believe by default, it's going to try to minimize this term. So it's going to try to essentially solve for x here in this case by randomly guessing. We spawn a new study using Optuna, and then we call optimize, and we pass in our objective function, and we give it the number of trials we want it to do. It's going to iterate through randomly guessing, and it's going to randomly converge to 2. So by randomly guessing, it got it right. And so that's what Optuna does. It's pretty easy to use, at least in terms of writing code. All right, so now that we have our model, we have our features that we've chosen, we know what hyperparameter optimization strategy we're doing. Now we can actually run the notebook and do the machine learning. All right, so out here, we've got our pipeline. So our machine learning pipeline, the first thing we're going to do to any sort of data that we feed in, we're going to standardize it so that it has mean of 0 and variance of 1. This is just generally a good practice with machine learning. And then we'll go into our XGBoost classifier. Our objective is going to be the binary logistic loss. It's going to be how we evaluate the model. And then down here, we actually have all of the hyperparameters that we're going to be searching over. So various things that I just thought would be interesting to look at based on reading the XGBoost documentation. So things like the number of trees in the model, a few regularization terms out here that help prevent overfitting, things of that nature. For each of these, I just pull from a different random distribution based on intuitively what it should be. So like, for example, in estimators, that's probably an integer. So we do that. We create the pipeline based on these params after randomly guessing, and then we score it based on using cross-validation here, I don't list it out here, but it's fourfold cross-validation using the logistic loss. And then we go ahead and so we've defined our objective per the previous slide. We then can create our study where we're trying to maximize the negative loss, so we're minimizing it. And so then we've created our study. And so then once I go ahead and do that, we can go ahead and go down and optimize. So that's what this code is doing here, where we give it our objective function. We tell it how many trials, so 128, and how many jobs. So this is why I ran this on the cluster, because there's 16 cores available. So I just gave this 16 jobs. And so then it kind of starts breezing through that. And we can go ahead and scroll down here. All right, and so here actually, as it's doing this hyperparameter optimization, is it's randomly choosing different things. What it'll do is it's actually printing out each trial. It's out of order here because this is in parallel, so there's multiple studies being run at the same time. And so this is useful to parse. What it does is it gives us the error of that trial. It tells us what parameters it tested, and then it says what the best it's found so far. And so then we go through and do that. And this is the actual hyperparameter optimization. So it's automatically tuning XGBoost for us using our training set. All right. So now that we have trained our model, I'll sort of interpret these error metrics later on. on a couple of slides later, but I'm just going to show generating these. So this is actually the log loss, as I'm showing out here, and it is the negative because the error of the model, we were using the negative log loss. So here we go. Then we can go ahead and evaluate the model against the testing set. So this is actually really important because the testing set is simulating brand new data that the model has never seen. It never influenced the model or its hyperparameters. So that's why it's important to hold out testing set. In this case, we actually held out like 50% of the training data. And only half of the data was used to train the model, just to get an idea of the error of the model. Then we train the model on the entire data set once we're confident it's not really overfitting. Here's an ROC curve that we get from that. And then we can go ahead and check a couple of specific cases. It's just sort of a sanity check for our model, where here we're going to take denatonium and glycerol. So denatonium, and there's actually a typo, we're looking at glycerol, not ethanol here. So denatonium is the most bitter substance that we know about. So it should probably predict that denatonium is bitter or something is very wrong. Glycerol is kind of sweet, so it should probably predict that it's sweet. And up there, we were just showing that it wasn't actually in the data set. The glycerol is in the data set. And so then we run these predictions, we append them to the array or to the data frame, and we say, okay, so denatonium is bitter, glycerol is non-bitter. So the model is making reasonable predictions. All right, and then we can look at the feature importances here. All right, so that's generating these plots. Now, what do these plots mean? So first, let's look at our test set performance. So the model performs very well on the testing set. It has a test set ROC AUC, so the area under the ROC curve, which is out here, of 0.898, which is good. So 0.5 would be random guessing, and then 1 would be a perfect model. Or I guess -1 would, yeah, and then -1 would be a perfectly imperfect model. Yeah. Anyways, so this metric essentially, we want it to be 1 for it to be a perfect model. Precision, so when we guess positive, how often is it really positive? How often is it bitter? Is about 76%. Recall, what percentage of bitter cases are we getting? Is 81%. Accuracy, how often are we right? Is 85%. So generally, it's doing pretty well on the testing set. So this gives us a hint that we're not overfitting to the data. Then we can look at the full data set. This is like training. The data has, the model has seen all of these examples at this point. That's why the AUC is very high, because it's perfectly predicting. Here is actually doing some cross-validation here. So we leave out a little bit of the data and train on it, and then we do that multiple times. So the cross-validated ROC AUC is about 0.8. And then the model, the evaluation metrics out here are similar to what we had from the testing set. So again, just showing that, hey, this works pretty well. And then finally, going out to what can, from assessing our model to getting some interpretability out of it, one of the important things about these tree-based ensembles, like gradient-boosted trees and XGBoost, is that we can actually get a little bit of interpretation out of it, like how important are some of these variables. So XGBoost can print variable importance scores. So here we have a little bar plot here showing that. So it turns out that the molecular weight is actually really important. It's the most important variable, at least in this data set, to binding. So which makes sense. The size needs to be right for it to bind to the receptor. H-bond donors are important. H-bond acceptors aren't important. So the molecule, if it has, or maybe I guess if it doesn't have, hydrogen bond donor, the presence of those hydrogen bond donors are actually important to binding, it turns out. The number of aromatic rings are also important. And then the other descriptors that we had weren't so important. And what this does is it gives us an idea of what characteristics of the molecule, if we had like a pipeline of molecules we wanted to check, what characteristics might be important. It also, if you wanted to go back and train a new model, this can help inform us of what other features might we want to look at. So maybe we want to look at the longest alkane length in the molecule to give a descriptor of like how long the molecule is. Maybe we want to look at like what the molecule size is spatially. Maybe we want to look at the acidity of the H bond donors, for example. So things of that nature. So we could go back and train a new model using those features and see if we get something better. So that's sort of the process we would take. And then one last thing, we're going to compare to the literature. So recall that our data set kind of seems to come from this paper here by Tuwani et al., which was this really deep and detailed paper discussing a bunch of different features, like thousands of features that might be relevant to detecting bitterness. So they look at a lot of different chemical fingerprints and things of that nature. It's a very in-depth discussion, and I would recommend checking it out. It's very interesting. So they looked at thousands of potential features. We looked at like 9 features just based on chemical intuition. They held out about 6% of their data as a testing set, judging by their SI. We held out 50% of the data as a testing set. They used a little bit of feature selection. We didn't, but it would be a good idea if we added more features. They did some dimensionality reduction. We didn't. But again, it's maybe a good idea once we add more features. They used 5-fold cross-validation. We used 4-fold. Not much of a difference there. They looked at 3 different models: random forest, ridge logistic regression, and XGBoost. We only looked at XGBoost. Their best models test set RC-AUC. So remember that 1 is the best. They got 0.892. Ours, we got 0.898. So just like, so comparable to the model, to the literature. And then their cross-validated RC-AUC is 0.86. Ours is 0.87, so it's comparable. So that's sort of, we're doing pretty well just with a couple of days of work on this problem. All right, so next up, we're going to go through, and now that we're done with the bitterness, we're going to talk about perovskites. Perovskites are really interesting. So a perovskite, there's a lot of work right now going on these for solar cell applications. Here is a good paper discussing that. Generally, a perovskite is of the form ABX3. Usually A and B, so here's the A site, here's the B site. A and B are typically metals, or at least species that can lose electrons easily. X is usually electronegative, so the oxygens here, these octahedra, these oxygens, those are the X's. And because these are really important structures, perovskites, there's actually a long history of trying to predict the structure. So one of the first attempts at doing this was actually Goldschmidt's tolerance factor from 1926. But this work still continues even to this day of trying to predict whether a material might form, or whether a composition might form a perovskite. Even up to 2019, where Bartel et al. Here actually had a tolerance factor that they developed using CISO. So what we're going to do is we're not going to be going nearly to the level of predicting structure. Instead, we're going to say, assuming it's a perovskite, what's going to be the volume per formula unit just by knowing the composition. So what A, B, and X are. So to do this, we need a data set. And what we're going to do is we're actually going to query the NOMAD repository. So NOMAD is actually a large database of computational chemistry calculations, including data from AFlow and Materials project, which is absolutely wonderful if we want to do some data science or machine learning on large amounts of chemical data. And so one of the data sets on their website is actually a set of stable inorganic perovskites. I'm not going to show the Jupyter notebook that we used in the interest of time. Instead, I'm going to show the code that we used, and we'll show a new pipeline on our platform that uses TPOT. So here I'm just formatting my search here using the NOMAD API. Check out their website, and there's some documentation on their API here. And essentially we're saying, go ahead and query the archive, and we're going to search for stable inorganic perovskites as the dataset name. And this will give us a bunch of search results. Excuse me. I then go ahead and convert those search results into ASE atoms objects. ASE is a chemical library, and it has a very convenient way of representing crystals. And we're going to use, and once we do that, we can then use XenonPy to generate descriptors. So XenonPy is actually out of this paper by Yamada et al., where it is a material science feature generation library. It's very nice. And so one thing it can do is it can take in the composition, or it can take in structure. We're going to be using compositional descriptors here. And so when it does a compositional descriptor, it's going to take a tabulated set of 58 different chemical properties. So things like number of s electrons in the atom, ionization potentials, electron affinities, things of that sort. And then it's going to combine the features in a variety of interesting ways, like weighted averages, standard deviations, minima, maxima, things like that. And all in all, this ends up with a total of 290 compositional descriptors for any given composition that we feed it in. So that's here, and here's the code where we do that. This is all going to be in the notebook examples on our API examples GitHub. And so we download XenonPy in this notebook, and one of the things that we have to do to set it up is we actually have to download the database of elemental data. So this only needs to be done once per installation, but we just kind of ran it here anyways. Then we spawn an element calculator out of XenonPy. This will take in a set of chemical symbols, which we're representing as a dictionary out here. And this will actually return us an array of a bunch of different features. And so that's kind of what we're doing here is we're featureizing our atoms out here. And then we're going to use TPOT. And so TPOT is an automated machine learning tool. So in contrast to the previous slide where we selected features, we knew the model we wanted to use, we knew the specific hyperparameters and their ranges, when we're running TPOT, we don't know that. We don't really care the model it chooses. We don't care how it combines the models it chooses. We just want something that'll predict. And so that's what TPOT does. TPOT uses a genetic algorithm to actually construct a set of transformations and models that'll do this prediction problem. And it can even generate code. So this code out here is actually Python code that TPOT wrote given an optimization problem. So it generates code. So that actually, I guess, puts this in the category of genetic programming. It would be, I guess, the category this is in. And what this does is it uses cross-validation to guard against overfitting, because think about it, this is a very, this is like the exact case where we would probably overfit, where the very model itself has a degree of freedom. So it uses cross-validation to help guard against that. It's very easy to choose the models it investigates. It has a lot of presets, including things like only allow models that are accelerated by the GPU, which would be potentially useful, or only look at neural networks, presets for classification, things of that nature. And so this is the thing we're using for automated machine learning. All right, so we're going to do that. And then we're going to go ahead and do some prototyping in Jupyter Lab. Here's the actual code to do TPOT. There's not a lot of code that's required. And I'll just walk through these real quick. Generations, we can tell it the number of generations that it's going to run for, or we can choose not to, and it'll just run until it runs out of time. We tell it the population size, so how many pipelines is it considering simultaneously. We can give it a max evaluation time in minutes. Here we're looking at 100 milliseconds. We can give it a maximum time in minutes. So here it's going to run for at most 5 minutes. We can tell it what type of cross-validation to use, the number of folds. Here we're going to use leave and out cross-validation because it works well with small data sets. We don't have that many examples in the training set. How verbose it is, what scoring metric it's using, one of these configuration dictionaries, which tells it what models it's allowed to use, the number of jobs. -1 just means run as many jobs as there are cores automatically, and then a random state for reproducibility. And then like any other Scikit-Learn model, we just tell it, hey, fit, here's our X and our Y, and it handles the rest behind the scenes. So very, very convenient to use. One other thing, by the way, note that we split our data. It might seem counterintuitive, but we have 144 examples. We're actually doing another 50-50 training and testing split here. So there's only about 70 or so examples in the training set. We're doing this because we have way more features than we have examples. We have like 290 features and 140 rows. If we fed that into into linear regression, we would always perfectly get a perfect model. So instead, we split it up into 50-50 training and testing, so that we really ensure that we get a good model. Because it'll probably be incorrect on the testing set if it is overfitting, in addition to this cross-validation that TPOT does to try to help guard against it. So this is so the training and testing split we're doing here, the really aggressive train test splits to try to help sort of sound the alarm if it's overfitting. But we don't actually, it doesn't look like we're overfitting though. Instead, here's the testing set where we held out 50% of the data. And then so it's on the diagonal, so it's pretty good. And here's the actual pipeline that it generates. So it's actually a very reasonable pipeline that we might get if we were creating a pipeline ourselves for this problem. So the first step in its pipeline is actually a feature selection based on this family-wise error metric, which is sort of saying based on a significance level and the number of examples in the or the number of columns in the data set, it calculates a cutoff for p-value of the descriptor relative to the thing that we're predicting, and it says if the p-value isn't at least this low, don't use this descriptor. So this is like eliminating some features, which is good because that's going to help combat overfitting, because the data set we're feeding in has more features than there are rows. And so this is going to help combat that. Next up, we do min-max scaling of the remaining features that we have in here. So again, a very reasonable choice that is pretty commonly seen in machine learning is scaling the data so it's on the same range. And then we get a linear support vector regression, which is also very reasonable. So support vector regression, it's going to try to minimize the sum of weights squared. And this is actually analogous to ridge regression, where there's a penalty term of the sum of weights squared applied to ridge regression. So, and ridge regression works really well in these situations where there's a lot of highly correlated features in the data set. And XenonPy generates a lot of highly correlated features. So ridge regression works really well here. It's analogous to linear SVR. So this is a very reasonable choice on TPOT's part. And so then we get this pipeline. We're pretty sure it's not overfitting. And then just to really hammer down that it's not overfitting, we're going to look at something that wasn't even in the data set from NOMAD, which is this samarium nickel oxide. So in our data set, there were only three rows entirely that had any F block elements, and they were all lanthanum. So the model has never even seen samarium. Nickel appears in the data set like twice. So it really hasn't really seen this crystal before. It hasn't ever even seen samarium. And so then we go ahead and we feed in the composition. The actual cell out here is four samariums, 4 nickels, 4 oxygens. We go ahead and generate features with XenonPy. We call our model, call its predict method here. Here's the known volume, and then we print it out. And so then the predicted volume is like 60 cubic angstroms per formula unit, because we actually normalized per formula unit. The known volume is 56. So having never even seen samarium, it is predicting the volume of the cell. So that's actually really, really interesting. All right, and so there's that. So we've sort of prototyped this. Now let's run this on the Exabyte platform. So what I did is I actually, on my user account, I created a machine learning workflow that uses TPOT. It downloads TPOT and runs it. And because I created this on my account, because our platform is very collaborative in nature, this is actually now available on the bank workflows. So anyone here can actually copy TPOT to their account and use it now. In order to use it, we'll upload a CSV containing some data. We'll, under important settings, we'll tell it the column to predict. We'll choose the training and testing split. We'll train the model, and then we'll use the trained model to make some predictions. And the last few slides in this webinar will go over that. So here's an overview of the TPOT workflow from my account. So we're showing this in my account's workflows. So here we go. Here is TPOT. So it looks like many of the other machine learning workflows that are on our platform. The platform was kind of busy the day that I recorded this, so it took a couple seconds to load. So here we go. This set up the job sub workflow. We'll just go ahead and set up the job as it says. It copies into the data set essentially. And then we have this machine learning sub workflow, which is out here. And so here's the parts of it. So we have some settings in here. We read in the data. We do our train test split. We run TPOT here. And then we create a parity plot to assess the model. And so then I can go over here and I can choose the model train and predict. And so then recall that TPOT is very easy to use, or at least it doesn't take too much code to use TPOT. And so here we actually just declare the TPOT model with some things out here from that I described a couple slides ago, the different parameters for the run. And that's really it. TPOT is now on the platform as a result of this because of our platform's collaborative nature. And so then there's that. Here's what it looks like. Here's the code that we used to have that. And then over here, we can actually go to the bank workflows tab, which I'll click here. So we can then go to the bank workflows and we'll see that it's actually available in there, ready to copy and use. And I'll go ahead and here it is, TPOT pipeline, May 24th, it's 7:28, and it's tagged AutoML. So there's the pipeline, or there's the TPOT. And so then to run a TPOT job, so here what I've done is instead of using my user account, I'm in a different user's account. I'm in the exabyte account. I can go to the bank workflows that we were just at, and I can find the TPOT pipeline that my account uploaded out here. I copy the workflow to my account. So here I'm on the exabyte account. Then now it's in my account, I can go ahead and create a new job. It'll load up. I can go ahead and give the job a friendly name so that I remember it weeks down the road when I'm looking at this again. I'm calling it TPOT webinar test. I can then select the workflow because I just copied it. It's like at the top of my workflows list. And I go ahead and click that. We see that we now have this dataset tab out here. So I need to select a dataset to show anything there. And I can actually find the Perovskite data that I have uploaded. Caveat, by the way, this particular, when we recorded this, we hadn't yet realized it's a good idea to scale the volume of the Perovskite based on the formula units. So here, this is actually just volume in cubic angstroms, not volume per formula unit. So here we can go to the machine learning sub-workflow and we can configure it a little bit. So here I tell it the target column name, which is volume. And then I can go ahead and edit TPOT to actually configure TPOT settings. So I go into the script and I find the settings in TPOT's initializer over here. So here I'm going to change it so that TPOT only runs for 5 minutes. And so here it's going to run for 5 minutes total, and then it'll stop. So that looks good. I'm going to scroll down to check the rest of this. All right. And then I close the workflow unit. And then I can go ahead and go up to the compute tab. And I can go down here and I can set the time limit. TPOT's going to run for 5 minutes, but it might take a couple minutes to download things. So I'm going to give it like 30 minutes. I'm going to put it on the Azure cluster this time. And so we have access to AWS and Azure, and I'm going to give it 8 cores. And then I'm going to go ahead and click the check mark to say, yep, this looks good. It'll appear in my account's jobs explorer, and then I can run the job, and then it's going to spin up the node, it's going to run the job, and it'll actually give me a pipeline. And here's the pipeline that we get from this. This is actually the parity plot that is on the results tab after the job completes. Note that I kind of took the TV chef approach here, where when I submitted the job, I had already actually trained another version just to get the recording done. And so this is actually already fit to the data. And we get generally good parity. So here there was 20% of the data was held out as a testing set. So here's the training set. In blue and green is the testing set. And so it looks pretty good. Interestingly, similar to our prototyping, we actually get a very similar and very reasonable pipeline from TPOT. We start off by scaling the data between 0 and 1, which is reasonable. Then we do some feature selection based on the p-value again, which is reasonable because we want to reduce the number of features to help prevent overfitting. And then we do ridge regression. Remember how a couple of slides ago I mentioned that the support vector regression was analogous to ridge regression? Well, this time it's actually picking ridge regression, probably because it works really well on these types of highly correlated feature spaces. And we get an RMSE after cross-validating of like 10.8. And here's how I actually knew the pipeline. It actually prints out to the standard out what's in the pipeline. So this min-max scalar selecting the percentile and then ridge cross-validation or cross-validated ridge. Yeah, and so here's how we can actually run predictions with the model that we just trained over here. So I can go ahead from a new job, I can select the workflow, which is this predict workflow that just got generated by TPOT. I can go ahead and choose my data set. I'm actually going to be a little bit lazy here, and I'm going to choose the Perovskite data set that I just uploaded, where I just want this volume. I just want to predict the volume here. Because I'm being lazy, I actually have to change the code a little bit, and this is actually demonstrating how easy it is to change the code. So in these jobs, we can actually change the code of the workflow without changing without changing the actual workflow in the bank or the workflow in our account. So this is just specific to this one job. And what I'm going to do here is I'm going to type out a comment for the purposes of this video. But essentially what I'm going to do here is I'm going to drop the volume column. Because right now our machine learning implementation kind of assumes that when we want to predict something, we don't know the column we're predicting. And so I'm just going to say, hey, because I'm being lazy, go ahead and just drop the volume column and run the prediction on it. Because the rest of the predict steps in our model are going, or in our workflow, are going to assume that the volume column is not there anymore because we're doing a prediction. We don't know it at this, we shouldn't know it at this point. I'm going to go ahead and give this workflow a friendly name, TPOT predict. Again, I'm going to go ahead and give it like 30 minutes, probably way longer than it needs, but it'll stop as soon as the job completes. This is just time for it to install packages and do anything else. Again, on Azure with 8 cores, I go ahead and click the check mark out here, and I can go ahead and once we get to our job explorer, I can submit the job. And here we go. We submit the job. All right, cool. And then finally, once this actually finishes, here's how I can get predictions. So here's how I got the parity plot from a couple of slides ago. I open up the predict job once it completes. I guess the parity plot was from the training anyways. And it'll print out the predictions, the row by row predictions that the model returns. And so here's that. I can then download the CSV. And so that's how we can actually now run TPOT on the Exabyte platform using the collaborative nature of it. So then just to summarize this webinar, because we went over a lot today. We ran Jupyter Lab on the Exabyte platform, which is great for prototyping. We featurized using RDKit and XenonPy. So RDKit's great for molecules. XenonPy is great for periodic systems. We learned how to train an XGBoost classifier, a very popular gradient boosting algorithm. We learned how to automate the machine learning process using TPOT. And then finally, we learned how to use TPOT as a workflow on the Exabyte platform. So finally, the next webinar is going to be next month. The code is going to be available on our GitHub from this webinar. We'll do a Q&A session in a second, but if any questions come up afterwards, please feel free to email us. And then finally, I'll just close out with a couple of very nice packages. RDKit is pretty good for generating features. So is OpenBabel. XenonPy is also good at that. Feel free to refer to these in the YouTube video afterwards. And then finally, some other machine learning packages here. Scikit-Learn is wonderful. We were using it throughout. For neural networks, TensorFlow and PyTorch are also very popular. And here's a couple of links for those out here. Alrighty, so thank you all. I'd be happy to answer any questions.
Thank you, James, for this informative explanation. There are no questions currently in the Q&A, and we're running a bit over time. So maybe let's take one quick question, if there are any. But let me start with a question from my side first. So when we're speaking about XGBoost, right, I think it was on slide 10 or around there. How do you select the objective?
The objective. So choosing, so like the objective that I'm feeding into Optuna.
No, I think let's go back to slide 10.
Slide 10, were we reading
maybe,
in the data set?
Slide 18. It was
18.
Cut at the bottom.
All right.
Yeah. So here, when you deal with it was 16. Sorry.
Okay.
So when you deal with setting up the parameters for XGBoost, right there, you have the objective as a parameter, and you currently have it as binary logistic. So maybe let's discuss how you make this selection.
Yeah. So I was just looking at different ways that, so I was sort of playing with XGBoost and just looking at different loss functions that it uses. Logistic loss is sort of good. It's not as interpretable, but it's good. It's good at like comparing between different models. So that's sort of the reason I used it. It was just an option available, and I was just testing it out. And it ended up working out fairly well. And then for these hyperparameters here, the way that I chose these and decided to tune these is I actually read XGBoost documentation. I read a few blog posts to talk about how other people had done this, because I was and still am learning how to use XGBoost. And so I just looked at what sort of hyperparameters are other people who are more experienced at this using. And it turns out that in estimators, these parameters here, the Greek letter coefficients out here are like, they help protect against overfitting. And there's a lot of different ranges. And again, these ranges here just come from looking at the XGBoost documentation and articles that other people had written about reasonable ranges to be searching for this.
Okay, so would you advise this to be a good first guess for someone who uses the same objective?
Yeah, I would say that this is probably reasonable. I mean, as always, I
Okay.
would recommend doing your own research, looking up, reading up on what other people are doing. But yeah, I mean, I would say that this is probably a fairly reasonable set of hyperparameters to be looking at.
All right. I don't think we have any questions, and we are a little bit over time. So with that, I think I'd like to thank you, James, and thank you everybody who participated today, all the attendees. We will be back next month, and our current plan is to talk about deep learning next month. So with that, thanks everybody again, and stay safe. We hope to see you again next month.
Thanks you all.